Today we will be discussing acute myotube protein synthesis regulation by IL-6 related cytokines. The first concept to explore involves a process governing the maintenance of skeletal muscle mass. This involves a balance between protein synthesis and degradation. An important class of cells, called cytokines, play a role in regulating protein structure and function, or proteostasis. Interleukin-6, or IL-6, cytokines are a family of cytokines that regulate important physiological processes involving the immune system, inflammation, and cell survival. Common targets for IL-6 cytokines are skeletal muscles, and skeletal muscles also serve as a source of IL-6 cytokines. Two important members of the IL-6 cytokine family are IL-6 and leukemia inhibitory factor, or LIF, which both play a role in skeletal muscle hypertrophy and atrophy. Hypertrophy is the enlargement of muscles, while atrophy is a wasting away of muscles, often due to degeneration of cells. Both IL-6 and LIF have the ability to induce intracellular signaling pathways via type 1 cytokine receptors. These receptors have alpha receptor subunits that bind to ligands and beta receptor subunits that transduce signals. Glycoprotein 130, or GP130, is a transmembrane protein that makes up the beta receptor subunit in IL-6 cytokines. When GP130 is phosphorylated, it leads to the activation of STAT3, or signal transducer and activator of transcription 3. STAT3 activation leads to target gene transcription and the induction of SOX3 expression. The expression of SOX3 can lead to suppression of GP130-dependent signaling, producing a negative feedback system. After GP130 influences intracellular signaling of the IL-6 family of cytokines, mTORC1 upstream regulators can be induced that increase protein synthesis. The induction of protein synthesis by STAT3 activation plays a role in hypertrophy of skeletal muscles. mTOR, also known as rapamycin, acts in the multiprotein complex mTORC1, which deals with skeletal muscle protein synthesis and regulation of muscle mass. The purpose of this study was to investigate if IL-6, or LIF, can regulate myotube protein synthesis through GP130-dependent signaling pathways. It is hypothesized that the acute administration of IL-6 or LIF will induce mTOR signaling and protein synthesis. Overall, the regulation of IL-6 or LIF on myotube-dependent signaling pathways was explored. There were 9 total steps in the design of this experiment. The first step involved performing a cell culture on C2-C12 myoblasts, where the cells incubated for either 4 or 8 days. The second step involved pre-treating myotubes with cell proliferation inhibitor prior to administering cytokines. The myotubes differentiated for 8 days. In the third step of the experiment, the myotubes were treated with different doses of IL-6 or LIF cytokines. RNA interference was then performed prior to intracellular signaling inhibition. A GP130-dependent STAT3 specific inhibitor was used to prevent the intracellular signaling of STAT3, which targets the mTOR signaling pathway. A Western blot analysis was then performed, and the myotube protein synthesis was determined using an incorporation of puromycin. The amount of synthesized protein was determined using a Western blot. RNA isolation was done prior to cDNA synthesis and real-time PCR. Statistical analyses was then done using a t-test, a one-way ANOVA, and a two-way ANOVA. This experiment concluded that IL-6 cytokines induce protein synthesis for doses of 10, 20, and 100 nanograms per milliliter, but not for a dose of 5 nanograms per milliliter. All doses of LIF induced protein synthesis. It was determined that both IL-6 and LIF induced protein synthesis at specific doses. To determine whether the myotube protein synthesis was a product of IL-6 family cytokine induction, the myotubes were treated with the cytokine that is not from the IL-6 cytokine family. No doses of this cytokine produced any change in myotube protein synthesis. concluding that the protein synthesis was a product of IL-6 family cytokine inductions. The figure that will be discussed is Figure 3, which analyzes the regulation of IL-6, or LIF, on myotube GP130-dependent signaling pathways. The C2-C12 myotubes were treated with IL-6 and LIF for time periods of 30 minutes, 1 hour, 2 hours, and 4 hours. In Part A of this figure, the phosphorylation of STAT3 and other signal transducers were measured using a Western blot. A Western blot is an analytical technique used in molecular biology that detects specific proteins in a sample. Often, antibodies are created that react with the target protein, and the sample then undergoes protein denaturation before the process of gel electrophoresis begins. After a series of washings, antibodies will allow visualization of the protein on the gel. In the wells analyzing STAT3 in this Western blot, introducing IL-6 cytokines produced less signal transduction than when introducing LIF, although both IL-6 family cytokines induced protein expression. In Part B of this figure, real-time PCR was used to measure the expression of SOX3 mRNA in myotubes treated with IL-6 and LIF cytokines over different time periods. The expression of SOX3 mRNA can be measured using optical density, which is represented on the y-axis in this figure. The expression of pSTAT3 upon induction of IL-6 and LIF cytokines can be seen to induce expression over all time periods, with the largest percent intensity of optical density being at 30 minutes and decreasing steadily for IL-6. The optical density decreases more rapidly for LIF, with the optical density dropping drastically at 2 hours. Part C of Figure 3 shows the SOX3 mRNA levels for IL-6 and LIF over 30 minutes, 1 hour, 2 hours, and 4 hours. Both IL-6 and LIF had the highest levels of expression over 1 hour. When each cytokine treatment is compared to the control, it can be seen that all levels of expression are greater, implying that cytokine induction has some effect on SOX3 mRNA expression and therefore also has an effect on the induction of protein synthesis. My discussion questions for today are 1. How do you think that IL-6 family cytokines increasing protein synthesis specifically causes muscle hypertrophy? And 2. Do you think that increasing protein synthesis using cytokines could affect other physiological processes within the body besides muscle hypertrophy and atrophy?